Hi students, once again I am meeting you to discuss more about what we have the lesson microbes in human welfare. And this category we have discussed a number of effects of microbes in human welfare. Now we have to talk about microbes as biocontrol agents. What is biocontrol? So it is nothing but actually eradicating harmful living organisms with the help of another living organism. That method is called biocontrol. Killing harmful living organism with the help of another living organism, that is called biocontrol method. Now here, so normally we are using, that one, pesticides and insecticides to kill the mosquitoes or any other insects or any other pest. So large scale applications of such chemical pesticides and then insecticides have deleterious effect on what is called human health. And also they pollute the environment. So in order to avoid such things, that is preventing that is what is called the effect of the fertilizers or any other pesticides on human health, and to prevent pollution, we are following what is called biocontrol method. What is biocontrol method? It is a method of controlling pests using microbes such as bacteria, fungi or viruses, or naturally occurring substances derived from plants and animals. Such method is called biocontrol method. Further, we are using some microorganisms. Such microorganisms used or any other agents used to control the pest is called a biopesticide. So normally we have organic pesticides, naphtha-based pesticides. They are all chemicals. They pollute the environment, causing damage to all the living organisms in this world. That's why we have biopesticide. What is biopesticide? See that one. The use of microbes or biocontrol agents to just control specific insect pest. Specific insect pest is called what is known as biopesticide. Control specific pest or insect pest. We can say specific pest, or aptly we can say insect pest. And that is called biopesticide. So two examples given in the book. One, beetle, what is called ladybird beetle, having red color, what is called the wings with a spot. And that one is used to control aphids. Aphids are nothing but the plant lice, just damaging the plants. They are being controlled with the help of what is called the ladybird beetle to control aphids, the plant lice. Another one, the dragonflies. They are used as what we have larvivorous insect, because they control mosquito larvae. They are feeding on mosquito larvae. That's why the dragonflies. So nothing but that one, the one which is coming before the rain, having a head abdomen and also long abdomen, head, thorax and long abdomen, with the two pairs of wings. Before the rain arrives, it indicates rain is going to come, and that is called the dragonflies coming under one order, Odonata. So the dragonflies are the best insect to control mosquito larvae. So these are all examples for biopesticide. We have to go for some more examples related to biopesticide. Weedicide or any other fungus, any other virus, or we have the bacterium, the Bt bacterium. I'll discuss one by one. Students, now let us discuss about the various examples as given in the book as biopesticides. Number one, Bacillus thuringiensis, the common bacterium which is used the best biopesticide, commonly called as Bt bacterium. So it's a soil-dwelling, free-living bacterium, not a parasite, which is commonly used as a biopesticide because it contains a toxin called a cryotoxin, CRY toxin, because it is produced by a gene CRY, hence called CRY toxin. Now scientists have normally introduce the gene, the cry gene. Different types of cry genes we have, CRY gene, into the plants. Have raised genetically engineered pest-resistant plants. Genetically engineered pest-resistant plants. One such plant which is resistant to pest is Bt cotton, as it is produced through incorporation of the genes, the Bt gene, also called by name Bt gene, from Bacillus thuringiensis, hence called Bt cotton. This is a genetically engineered plant which is highly resistant to pest, insect pest. Now, what is happening? What is the effect of this one? How far it is useful to control the pest? So, during sporulation, now this bacterium produces a crystal protein. The name of the crystal protein is called delta endotoxin. This is a protein, delta endotoxin. This is a toxin poisonous to insects. And this is produced under the control of a gene, what is called CRY gene. We have different types of genes under this crime. So, here CRY1AC, this is one type of gene. CRY2AB, another type of genes. These genes are responsible for the production of toxin to just work against the cotton bollworm, the one which produces abnormal cotton, that is what's called boll, and that bollworm is controlled by the genes, these two genes, CRY1AC, CRY2AB. They produce a toxin. They kill what is called the bollworm, which destroyed the cottons. Another cry gene, CRY1AB, and this is actually a gene which encodes for a protein. That protein toxin kills the corn borer, the one which affects what is called the yield of corn. So these are all the neat questions. So we have different CRY genes for different organisms. Their effect is to produce a toxin to kill a particular insect pest. Okay. So these toxins have specific activities produced by different genes. Here these toxins have specific activities against insects of orders. We have 29 orders in the case of insect, what we call the class Insecta. Now these toxins are working against some of the orders, including the insects, or they kill the insects of the orders. Here in your book given there are 4 orders. These 4 orders include different types of insects. These insects are controlled by what we call the delta endotoxin. Order number 1, Lepidoptera. Normally it includes, you know that one, the moth and butterflies. Here this Lepidoptera includes the main insect which is being controlled by delta endotoxin is tobacco butworm and armyworm. These are the 2 worms commonly destroy what we call the plant tobacco. So they are being controlled by what we call delta endotoxin. The second order, which includes the beetles, Coleoptera. So the beetles are controlled by this delta endotoxin. The third one, Diptera, which includes the mosquito and flies, including the house flies also included under this order. So Diptera meaning having only one pair of wings, unlike 2 pairs of wings in the case of insects. So Diptera includes mosquito and flies that are being controlled by this toxin, delta endotoxin. The last one, Hymenoptera, the one which includes bees and wasps. Honeybee is also included under this order, Hymenoptera. So some bees which are harmful to the plants, acting as a pest, they are being killed by this delta endotoxin, and these bees are included under order Hymenoptera. So this delta endotoxin is highly effective against insects, those insects which are mainly included under these 4 orders. So this is about one, that is Bacillus thuringiensis. How far it works, how it kills the worm, that is particularly the caterpillar it kills by working a specific mechanism. So what is the mechanism adapted by this Bt bacterium through its toxin to kill the caterpillar larva of insects? Let's see now. Now let's see the mechanism of action of Milky bacterium. How it kills the larvae, the caterpillar of an insect of the orders what I mentioned. Now this is the BD bacterium which produces a crystal protein which is insoluble. So when the insects, you see that one ingests the crystal proteins. Now here the available pH is 12. High pH is available here, pH here 12. That is alkaline pH. Alkaline pH. So strongly alkaline pH. So the alkaline digestive tract denatures. So first one ingestion and then solubilization, denatures, converted into what we have the monomer and polymer. Converted monomer, then accumulated together to form polymer. So the solubilization is nothing but the denaturation of the crystals. Now what is happening? The insoluble crystal toxin making them soluble. So it's been converted into what is called soluble toxin. That is what is called solubilization. Then the cryotoxin is activated. How? So this cryotoxin then gets inserted to the membrane of the midgut cell. So there is a binding receptor to which what we have actually that is the toxins are bound to the surface of the midgut cells. This is the midgut cells. You see that one, that is activation, that is being transported. Normally here I just explain like that when I drew the diagram, but actually here the cell is present lining the midgut. So activation means nothing but actually there is a toxins, the soluble toxins are bound to the receptor present on the surface of the midgut cells. The next one is being converted to toxin monomer, toxin polymer, and they get inserted into the membrane of the midgut cell and creating the pores. Through the pores, what will happen? The water enters, the swelling of the cells occur and causes the osmotic lysis, the breakdown of the cells, which results in the death of the cell. This is what is happening. So the cryotoxin then gets inserted in the gut cell membrane and paralyzes the digestive tract by causing openings. Through these openings, we have the entry of water leading to the osmotic lysis of the cells. So the midgut becomes normally paralyzed. So the animal is unable to eat. So stops eating process and just as a result stop to death. That's the result. The death of the individual occurs because of starving process. Because the animal is unable to take the food. That causes actually the starving of the animal, particularly the larva, leading to the death of the individual. So production of toxin by the bacterium, there is a crystal protein. It is being ingested, then it is being actually made into soluble substance, then it is being activated by way of binding to the receptor, converting them into toxin monomer, toxin polymer. Then membrane insertion occurs. Normally the toxin gets inserted into the cell through the gut cell membrane, and there occur the formation of pores through which water enters, causing the swelling of the cells and the bursting of the cells, leading to the cell death. This is the mechanism of BD, what is called toxin released by the BD bacterium. Let's see 2 or 3 more what we have the biopesticides. Some small things, for example, BD sites, some examples of biopesticides. Weeds are plants that affect the growth of normal plants by utilizing all the nutrients available in the soil. So to control the weed, we are using what is called weedicides. So weedicides are substances that destroy the weeds without actually harming the normal plants. Without harming the normal plants, we can say the useful crop plants. So useful crop plants. Then bioweedicides are nothing but substances, or we can say secondary metabolites, or secondary metabolites. So we can have primary metabolites and secondary metabolites are products of metabolism. Primary metabolites are useful to the possessor, the one which produces. For example, hormone is a primary metabolite. Then if you are taking antibiotics, it is a secondary metabolite. It is not useful to the producer but useful to others. That is called secondary metabolite. So bioweedicides are substances or secondary metabolites derived from either bacteria or fungi or from protozoa. Now, the first bioherbicide developed in 1981 It was a mycoherbicide produced from a fungus, what is called Phytophthora carnivora. That's the name of the fungus from which we have derived what is called the first mycoherbicide. Now, what is its role? It controls the growth of strangler vine. It's nothing but a milkweed growing on the trees of what is called the citrus plants, then just affecting their growth. So this weedicide is used to kill what is called strangler vine, a kind of milkweed growing on the surface of the tree trunks of what we call the citrus fruits, thereby affecting their growth. So we can control this strangler vine with the help of that is what is called the weedicide that is obtained from Phytophthora carnivora. It is a myco, that is what is called herbicide because it is obtained from a fungus. That's why it's called mycoherbicide. Okay, that's about the role of weedicides. And two more we have, that is the end, we have to conclude. Some viruses and also some fungus. Okay, what is the role? How far they are working as a biocontrol agent. Let's see now next. The next biopesticide organism is Trichoderma. It's a free-living fungus, normally found in the root ecosystem. It is being used as what is called a biocontrol agent against many what is called plant pathogens, the one which cause disease to the plants. That is called plant pathogen. So many plant pathogens are being destroyed with the help of what is called Trichoderma, a free-living fungus. Now the next one, Bacterovirus. In the book it is given Bucloviruses, it is not that one, it's a Bacterovirus. So it acts as pathogens and attack many insects and also other arthropods, coming under the category Arthropoda. One such genus of Buclovirus is Nucleopolyhedrovirus. It is a genus of what is called Bacteroviruses, one type. Now, these nucleopolyhedroviruses are also used as biocontrol agent. These viruses are highly species specific and have a narrow spectrum of insecticidal applications. They have no broad spectrum of killing the insects. They are species specific and they kill only definite number, a specific number, a specific what is called the insects. That's why I mentioned here narrow spectrum of insecticidal properties. So this is all about what we call the microbes as biocontrol agents. We have to proceed what is called actually the microbes as biofertilizers in the next class. Okay, thank you.